Thank you so much for the introduction, and hi everyone. Yes, let's just get right into it. Today we're going to be talking about microbiome evolution within the honeybee. And so perhaps you're like me, you like eating food, and it may come to a surprise to you, maybe not, that over 30% of the food that we eat, that is the crops that we rely on, these crops are dependent upon pollination and services by pollinators. But if we look at this plot, you may have heard something called colony collapse disorder, where mysteriously honeybee colonies just vanish or die at incredibly high rates relative to what we saw in the 1990s and 1980s. And in this plot, on the x-axis, we have time, and on the y-axis, we have some proportion of loss. And I want you to look at the red line, which is essentially this sort of proportion of colonies lost in all beekeepers in North America in winter. And you can see that over the past decade, this number has continued to rise and has reached really, really high levels. I believe in the past year, something like 63% of honeybee colonies died during the winter. And so there's a big question about what kind of things are causing this and what can we do to sort of solve this problem to maintain the sort of our needs by these essential pollinators. And so multiple stressors actually contribute to the loss. It's more of a syndrome rather than a specific disease. But we know that the microbiome buffers negative effects from almost all these stressors. Bee-associated microbes protect against fungal pathogens. Some microbes have been shown to actually supplement nutrition via amino acid provisioning. And these microbes have been shown to sort of metabolize and sort of buffer against pesticide toxicity. And so nearly any sort of stress that you can think of in the honeybee colony world, bacteria have been shown to sort of ameliorate the effects, which is really, really surprising. And so despite these important host-relevant phenotypes that the microbes sort of confer upon honeybee hosts, we know actually very little about variation within natural microbial communities, especially during seasonal variation when bees are most vulnerable. And so sort of the primary question of this talk and kind of what I'm trying to get through in this current chapter of my thesis is, does evolution occur within microbiomes of honeybees? And I'm going to hope to convince you today that it does. Of course, you may ask about ecology, and ecology happens too. I'd love to talk about that. But today, in the interest of time, we're going to focus explicitly on evolution. So in this talk, I'm going to show you three things. I'm going to walk through the experimental design, sort of what we're working with, what kind of bacteria we're interested in. I want to provide evidence of allelic sweeps in rapid evolution. And then finally, I'm going to show, I hope that convince you that phylogenetically distant genera within the honeybee microbiome share similar functional responses in these allelic sweeps. Okay, and so what I did in the past couple of years is that I followed 12 honeybee colonies, found in 3 different apiaries split across 2 states, on average, about 300 kilometers apart. And I sampled these monthly from April 2023 to March 2024, or until the colony died, usually in winter. At each sampling, I took 60 individual workers, which I then split into 3 pools. So I now have 3 technical replicates, each consisting of 20 individual workers. I homogenized and pelleted them. I also sampled larvae, one of the brood casts that develop into adult bees, but we're not going to talk about that. And this kind of sampling design allows me to compare sort of how genetic variation of the microbiome changes over each time point within a single colony, and then compare this against different groups of sort of sampling structure. So I can compare colonies between colonies, apiaries against apiaries, that sort of thing, and look at sort of different batch effects from the sampling. But for now, we're going to look at individual colonies and how they change over time. So overall, we recovered a high number of metagenomically assembled genomes, or MAGs. Excuse the title. I assembled over 300 metagenomes by the end of this study and recovered on about 1,700 actual MAGs. After quality filtering, so kind of removing MAGs that fall below a certain contamination and completion threshold, we retained a little over 1,000 of these genomes. And here you can kind of see 2 different relative abundance plots of these individual MAGs. So within a single honeybee colony. So on the top, these are on the top plots here on the right. We have a genus of bacteria called Bifidobacterium, and on the bottom we have another genus of bacteria called Bartonella. And the different colors within the respective plots correspond to the abundances of individual bacteria. mags. And you can kind of see that overall that there might be some variation, especially in the bottom plot, and stochasticity about how these abundances change over time, but overall the colors are fairly stable. And this pattern holds actually when we look at abundances at a higher taxonomic level. So same colors, but I'm now looking at the level of genus, so a level up from the mags. And we can see that overall these communities, the colors that I want you to look at, don't want you to be necessarily interested in the taxa, but the colors themselves stay fairly stable, and don't really change much, with the exception of this middle colony, which I'd be happy to talk more about, but it has to do with the onset of winter. Okay, so I want you have to take my word for it, but I want these mags are stable with respect to relative abundance and have ecological communities respond weakly to seasonal change. And so again, I'm happy to talk about ecology at a later time, but overall that these mags don't really respond themselves much strength with changes in seasonal variation. So next, I'm going to walk through some allelic sweeps and rapid evolution. And so what I'm looking for are what are called allelic sweeps as a consequence of selective sweeps. And if you're not necessarily familiar with that is, or you need kind of a quick review, you can imagine I have a population of bacteria with high genetic diversity. And so on here, each one of these little brown bacteria are individuals within a population, and each one of the colored balls or little orbs correspond to mutations within that bacteria. And after a selective sweep event, maybe an antibiotic or some sort of like strong abiotic interaction, what this does is it selects for a certain mutation or variant that, and it purges the diversity at that location. And so if I look at that same location at a different time point, we see that within this dotted box that all of the mutations in this area have been replaced by a single sort of set of mutations. And so the diversity within that region is much lower than we might expect. And so these sort of like characteristics are sort of patterns of what we're looking for. And these are sort of nice, really good examples of potential adaptive evolution. And so I find candidate sweeps by looking for rapid changes in allele frequency. And so here's a representative example. On the top, we have two little plots. On the top, we have the relative abundance of this particular mag over time. And on the bottom, we have this sort of relative frequency of each of the alleles that I'm tracking within this bacteria. And so this goes from 0 to 1. If you see, I have two different colors here, and I want you to pay attention to the lines in blue, which are the alleles that I have identified to be rapidly changing. Rapidly changing means it goes from like 20% allele frequency or less to over 80%. So over a 50% change in allele frequency between any two time points. And the little colored rectangles in the background, the yellow, orange, and blue, correspond to the month when there is a change in the season. And so in orange is when the onset of fall, and blue is the onset of winter. And as you can see, throughout the time point, the alleles don't really change in frequency until the onset of winter. And then we have this huge, large peak corresponding to a rapid change in allele frequency across about 25 different genes in this bacteria. Now, what's interesting, you might be saying, well, Chris, that might just be ecology. Maybe this thing is being replaced by another bacteria. And so this circle plot represents alleles fixed before colonization. And so you can imagine we have that same bacteria, and we identify alleles that are fixed at 100% frequency at the first time point. So these are alleles when I first look at this bacteria, I see a bunch of mutations that are fixed at 100% frequency. And these are likely mutations that were already fixed on the bacteria before it even entered the honeybee colony. And then as we sample it, we see that new alleles appear over time. And so the circle plot represents a proportion of the start alleles that are no longer fixed, meaning that the start alleles were at 100% when I first observed the bacteria. And by the end of the experiment, or at some time in between, those alleles went down in frequency, like below 90%. And so when if there's a lot of blue in this circle plot, that means that a lot of those original mutations that were fixed are no longer fixed anymore. And that might be evidence for an ecological process, like invasion by another strain. And here's another example that I think really might illustrate that really well. And so this is a bacteria belonging to Lactobacillus. And you can see that in that circle plot, that about 75% of that plot, those original mutations that were there at high frequency have been replaced by something else. And you can kind of see that throughout, maybe make sense of what happens is that as we're going along on the time series here, we see that this huge change in allele frequencies around the onset of spring, and then it goes back down and then slowly rises again, kind of corresponding neatly with the sort of shape of the relative abundance trajectory, and the sort of rapid changes in the allele frequencies looks like something like a genome-wide sweep, where all of the mutation in one bacteria, all the alleles were completely removed, and then it slowly recovered and potentially even displaced the other one. And so there's lots of evidence for rapid evolution with interesting sweep trajectories, and sort of like a panel of the different sort of patterns we're seeing. So I've provided evidence, I hope, of allelic sweeps and rapid evolution. And now I want to convince you that, that these are happening in distant genera and share similar functional responses. I'm kind of interested, what are the targets of these sweeps? And so we're looking at sweeps that occur in positions where any variation causes an amino acid change. These are non-synonymous sites, a part of the codon where if you have any mutation, it results in a change in the codon. And this is an intersection plot that's basically counting the number of these types of sweeps that we observe. And we see that there's sort of a heterogeneous distribution by taxa. So these are different core genera within the honeybee microbiome. And we see something like Gillimellis gets the most, while something like Bombolactobacillus doesn't get very many. But what's interesting is that genera show convergent responses to seasonal change. So if you see an intersection between any of these different genera, that means that in the same month, so between May or June or July, the same sort of functional protein is being hit by one of these non-synonymous adaptive sweeps at the same time. And so we see that a lot of these sort of like targets are actually being hit by these sweeps as these bacteria all converge on the same sort of seasonal variation. To me, suggesting potentially that these illustrate sort of like targets of convergent response in response to the change in season. And with a statistical model, we can actually test that these parallel sweeps happen more often by chance. Maybe this is just sort of random, and we're just observing sort of the random distribution of these things that happen to be shared. And so on the y-axis, we have the axes represent log-scaled enrichment of the sweep count. That's the x-axis. And then the non-synonymous mutation count, so the y-axis, relative to a null model. And back in the sort of dots that are on the top right of this quadrant are genes that are enriched for both non-synonymous mutations. So these are these sweeps, and genes that are enriched for parallel non-synonymous sweeps. And so to interpret this, you might say that in this quadrant are genes that sweep in multiple taxa are also acquiring more synonymous mutations than you might expect. So not only are they being hit more often in multiple genera, but they're also being mutated more often within the genera that they're found. And so I hope I've convinced you today that evolution is happening in honeybee microbiomes, and it happens quite rapidly. This whole experiment was done over the course of 9 months. But I hope to have shown you that bacterial species within honeybees are stable over time, that these species exhibit rapid changes in allele frequencies, often concurrent with changes in seasonal variation. And then finally, rapid changes occur in similar functions across disparate taxa, suggesting convergent responses to seasonal variation. And with that, I will thank everyone for your attention. This has been an invitation. This is a great opportunity, and I'm very happy to share my work and hear your questions. Thank you.